This is a review video for mitosis and meiosis. Actively dividing eukaryotic cells pass through a series of stages known collectively as the cell cycle, 2 gap phases and an S phase, in which the genetic material is duplicated. In the M phase, the process of cell division occurs, and you have a nuclear division called karyokinesis, followed by the separation of the cell cytoplasm, also known as cytokinesis. This produces 2 identical daughter cells. Chromosomes are a key part of the process that ensures DNA is accurately copied and distributed and appears only during the M phase. They are a condensed package of DNA. Recall that during the S phase, DNA is replicated, and then it is wrapped around the histone proteins to form nucleosomes. Nucleosomes are then coiled up and stacked together to form fibers known as chromatin. During the M phase, at the very beginning, the chromatin must be greatly condensed into the chromosomes. Each chromosome is made up of 2 identical sister chromatids held together at the centromere. The centromere then appears as a constricted region on the chromosome. It serves as an attachment point for the spindle microtubules. A multi-protein complex called the kinetochore assembles at the centromere, and later the spindle microtubules will attach to the kinetochore. Cells that carry 2 sets of chromosomes are called diploid, also known as 2N. N refers to the chromosome number. The 2 chromosomes of a homologous pair are alike in structure and size, and each carries genetic information for the same set of hereditary characteristics, the same genes. However, keep in mind that the alleles of each gene need not be identical. Mitosis is the part of the cell cycle in which sister chromatids separate, and the cell undergoes division. During metaphase of mitosis, the chromosomes align up randomly in the center. Then during anaphase, the sister chromatids will separate, thanks to the action of the spindle apparatus, pulling them apart. From a single cell, mitosis produces 2 cells that contain the same chromosomal information. The resulting daughter cells are genetically identical with each other and with the parent cell. Both new daughter cells contain a full complement of chromosomes. Both are diploid, or 2N. Meiosis is similar, if you look at it, but very different than mitosis. The purpose for meiosis is sexual reproduction. Due to crossing over and 2 divisions, meiosis produces haploid gametes with unique combinations of maternally and paternally derived chromosomes. The first phase is known as the reductional division phase. That's because the number of centromeres, each representing 1 chromosome, is reduced by half. Components of each tetrad separate, yielding 2 dyads. In prophase 1 of meiosis, chromosomes condense and homologous chromosomes pair up and synapse so that crossing over can occur. Chiasmata are formed between homologous chromosomes at sites where genes are the same. An entire segment of each chromosome can be exchanged. This is called a swap. This exchange is reciprocal, such that each chromosome gets the same region of the chromosome segment from the other parent that it donated to the other chromosome. Crossing over between non-sister chromatids is what creates genetic variation, and this is why we call it recombination. The homologous pairs line up randomly on the metaphase plate during metaphase 1. And in anaphase 1, the 2 chromosomes of a homologous pair will separate, again thanks to the spindle apparatus. Then telophase 1 results in 2 cells having half the original number of chromosomes. Again, that was the reductional part. For the second part of meiosis, M2, this is the equational division. During the second metaphase, the individual chromosomes will line up on the metaphase plate randomly. Then during anaphase, the sister chromatids will actually separate. The centromeres divide, and they then move apart as chromosomes now to the individual poles. The result of this is 4 haploid cells that are genetically unique because of crossing over between non-sister chromatids, and again, the fact that those homologous pairs lined up randomly. I hope this helps you better understand the difference between mitosis, which is for the production of genetically identical cells that will replace old cells or allow an organism to grow, and meiosis, that is important for making the sex cells, also known as gametes, that are haploid and genetically unique. Thanks.